Hello to everybody. This is Mahdi Mohakik. I hold PhD of Anatomical Sciences from Tabriz Mudarris University in Tehran, Iran, and recently I finished my research fellowship in male infertility at Wake Forest University in North Carolina in the United States. Uh, I'm gonna present my research project entitled "The Mouse Testis Tissue Culture Could Resume Spermatogenesis as Same as In Vivo Condition After Human Spermatogonial Stem Cell Transplantation." It is a part of my PhD thesis in Tabriz Mudarris University that I presenting for you. As an introduction, as you know, the spermatogenetic process is a complex process which started with spermatogonial stem cell at the base of seminiferous tubule on the basic membrane of seminiferous tubule that it will continue with mitotic and divisions that some morphological change named spermiogenesis, and the last stage is spermation that will release the sperm in the lumen of seminiferous tubule. This spermatogenetic process is under the effect of different factors such as hormones, some growth factor interaction between this cell complex in the seminiferous tubule, that if there is any problem in any of these factors could be resulted to different level of infertility. But one of our choice to keep the fertility of our patient, especially that peripubertal boys that need to some gonadotoxic treatment of their cancer, is isolation, culture, cryopreservation, and transplantation of their spermatogonial stem cell. And it could help to avoid returning the cancerous cells to their body after treatment of cancer. Uh, we can induce the spermatogenesis by our reserved spermatogonial stem cells by two-way: in vitro spermatogenesis and in vivo spermatogenesis. In vivo spermatogenesis, we can transplant the spermatogonial stem cell to the host testis by in vivo condition, and in vitro spermatogenesis, we can transplant the spermatogonial stem cell to the host testis in the laboratory space and out of the host body. In vivo spermatogenesis, in vivo transplantation of spermatogonial stem cell has done by different researchers in different species in different years, from 1994 by Brinson and Zimmerman, and also it's continued by different researcher in different year, and it was very successfully, but just in rodent and some mammalian host. But our discourse will be focused on in vitro transplantation. In abbreviation, it should be IVT of spermatogonial stem cell. The IVT has a lot of advantages, include we can observe the transplanted cells in every day, in every week that we need, and it's very useful for study the behavior of spermatogonial stem cell in details, such as how they can homing on their niche, how they can progress the spermatogenesis, and how they can participate in the spermatogenesis complex. And also we can use this method to circumvent the immunological rejection or any other effect that should be happen in the host body and tissue if we want to in vivo transplant. But in vitro transplant, we can circumvent this effect. The objective of this study is we're gonna examine the progression of spermatogenesis after transplantation of spermatogonial stem cells under condition of testicular tissue culture in comparison with in vivo condition. So our methodology should be in this diagram that we have different phases. In first was our we got a human test sample from some other sperm non-obstructive other spermia patients that refer to Gandhi Hospital in Tehran, Iran, in IVF ward. After freezing and thawed the human test cell, we isolated the spermatogonial stem cell and then we confirmed the identification of these cells by tracing PLZF protein. As you know, PLZF protein is a specific protein for stem cells, not for spermatogonial stem cells, but it's specific for stem cells that we just we're gonna make sure that this cell pool is uh, contain some spermatogonial stem cell that has stem cell characterization. And we cultured these cells for 2 weeks. After that, we labeled that with DII staining. On the other hand, we prepare adult mouse with injection of 40 milligram per kg busulfan by intraperitoneal injection to make azoospermia mouse model, and we confirmed this model by H&E staining of testis. And I will show you that the result of the confirmation of our azoospermia model. And then after that, we isolate the testis of this azoospermia model for next step. In the uh, third step, we had two group in the test group and control group. In test group, we transplanted the spermatogonial stem cell by in vitro transplantation to the host testis in out of the mouse body, and in control group we transplanted the spermatogonial stem cell by in vivo transplantation in the host body. After 8 weeks, I'm gonna remind that we put the host testis tissue after in vitro transplantation on the agar gel for tissue culture condition. It's very important. We're gonna make sure that every procedure is out of the host body, and it's the difference between in vivo and in vitro transplantation. After 8 weeks, that is the period of spermatogenetic process in human, because our source, our spermatogonic stem cell source is human. We did our characterization studies such as histological studies, H&E staining, morphometric studies, tracing of DII, and also immunohistochemistry studies and molecular. We collected the test sample from a spermia patient in Gandhi Hospital with age between 20 and 60 years old, and we have some inclusion and exclusion criteria, and all of patient information is achieved and archived. As you know, as you can see, there is a result of confirmation of our other spermia mouse testis model. In picture 1 and 3, you can see the azoospermia mouse model, and 2 and 4 is control model. And you can see after injection of 40 milligram per kg busulfan by intraperitoneal injection to mouse body, you can see that seminal fistula is fully empty, and this just remaining of some Sertoli cells, and it's very important to lose the epithelium in the seminal fistula in comparison with control. And also, we confirm the identification of our isolated cells by tracing the pairs of the protein, the specific protein for stem cells. In the picture 1, you can see the red result, it's related to PSF protein in colonies derived from spermatogonial stem cell culture is first contrast, and the picture third is the negative result without a primary antibody. And as you can see, the picture one is related to in vitro transplantation of the spermatogonia stem cell to the host testis in out of the body, and the picture two is in vivo transplantation in the body of host mouse, and you can see the filled seminal fluid stool of host testis by transplanted cells. And there is a result of our host testis after 8 weeks. It's this slide is regarding to H&E staining. You can see the picture one and two is related to IVT group in vitro transplantation after 8 weeks. You can see the spermatogonial cells, some spermatocyte, and maybe because of condensance and some elongated nucleus of these cells, it should be long spermatid or spermatozoa. And in the H&E staining, we cannot certify these cells by H&E, just we guess that it should be these cells. And figures number 3 and 4 in vivo group, you can see how the spermatogenesis is resumed and complete, maybe. And picture 5 and 6 is related to control group without any transplantation without transplantation. And the result of immunohistochemistry studies is in this slide. You can see these three proteins, PLZF, SCP3, and ACRBP. PLZF, as you know, is a stem cell factor, and it's specific for stem cells. SCP3 is a protein specific for spermatocyte, and ACRBP is for long spermatid or spermatozoa. As you can see, the green figures is related to our specific proteins. The red one is the tracing of DII, which is stained at the first day of transplantation, and it need to trace at the same time with our antibody. And the blue one is the tracing DAPI staining, that this is a cell, is very important. And the last image is related to merge of these three figures together to show that indicated cells is correct. And this slide is related to in vitro transplantation group, and this slide is related to in vivo transplantation group. You can see same as the last slide, the tracing of PLZF, SCP3, and ACRBP protein, tracing DII, tracing DAPI, and merge of these three figures together. And in morphometric studies of host testis after 8 weeks after transplantation, you can see the counting the number of different cells in seminal fluid tubule include spermatogonial stem cell, spermatocytes, and sperm-like cells. That there is a significant difference between in vivo transplantation group and in vitro transplantation group, but the result is very close together. And also there is a significant difference between in vivo and in vitro transplantation. Also, the result of percentage of tubules with epithelium, there is also the significant difference between these two groups, in vitro transplantation and in vivo transplantation, but as same as the last result, this result is very close together It could show that it's very near to get this achievement to reach out the in vivo condition. The result of real-time PCR just show that in PRZF, TECTA1, and TMP1, this gene is respectively related to spermatogonial stem cell, spermatozoite, and sperm-like cell or spermatozoa. But there is no difference, significant difference between the two groups in expression of these genes. In different groups include IVT and in vivo transplantation. The conclusion of this study should be uh, in vitro transplantation can support spermatogenesis induction by spermatogonial stem cell transplantation as same as in vivo condition. Thank you for your attention, and I will be available if you have any question, and I can answer that. Thank you. Thank you. Have a good day.